Good afternoon, everyone. I am your host, Shailesh Shukla. We will be starting the webinar very shortly. Thank you for your patience. We will be starting the webinar very shortly. Thank you for your patience. Good afternoon, everyone. I hope you are doing well. On behalf of Advent Informatics, I, Shailesh Shukla, warmly welcome you to today's webinar on a highly innovative and practical topic, which is unlocking the innovative modeling of PROTAC using Molsoft tools. We are delighted to have you with us and look forward to an engaging session filled with the knowledge sharing and real-world applications. Before we move into today's session, let me take a moment to introduce the organization behind this event, which is Advent Informatics Private Limited, headquartered in Pune. Advent Informatics is a leading distributor of advanced life science software solutions. Our expertise spans chemoinformatics, molecular modeling, medicinal chemistry, chemical data curation, and biological information management. We proudly serve as a wide range of clients, from leading pharmaceutical and chemical companies to CROs, academic institutions, and government R&D lab across India. Our mission is simple yet impactful, which is to bridge the gap between scientific challenges and software solutions by making world-class technology accessible to Indian researchers and professionals. Advent Informatics is honored to represent globally renowned technologies partner including Molsoft, Chemaxon, Campass, Satara, Transgene IT Solution, and Key Module. With their support, we continue to empower discovery, innovation, and collaboration in the life science domains. It's now my absolute pleasure to introduce today's speaker, Dr. Shamshair Singh. Dr. Singh is an expert in computer-aided drug design with over 20 years of pharmaceutical and life sciences industry. Currently, he serves as a senior application scientist at Advent Informatic here in Pune. His deep expertise and practical insights makes him the perfect guide for today's session on unlocking the innovative modeling of PROTAC using Molsoft tools. So please join me in welcoming Dr. Shamshair Singh. Sir, the floor is yours.
Yeah, thanks. Thanks, Sailesh, for such a nice introduction. So, are you able to see my screen?
Yes, sir.
Fine. That's great. Hi, good afternoon, everyone. So, as thanks for joining, and today's, as you know, everyone is very much interested in understanding how the uh, PROTAC is, how the PROTAC works, and how can we model the newly designed PROTACs using Molsoft tools. So this is today's topic. What we have is unlocking the innovative modeling of PROTACs using Molsoft tools. And this is something about our company. So we are Adventate based out. We were founded in the year 2016 and based at Pune. So what do we do is we provide, we are a distributor of molecular modeling, bioinformatics, and other chemoinformatics tools. So this was the survey which we ran last year in the month of November, for which we were highly appreciated by our clients. Like in less than 1 hour, we could respond to your queries in up to 50%, and less than 3 hours, we could respond to your queries to 85%, and in less than 24 hours, we could respond to your queries in less than, 100%. So we are the distributors, not the developers of these softwares. Again, and let's assume, like, if you face any kind of issue with regards to the any of these tools which we provide, like the way we follow the process here is, like, once we receive an email, the best possible thing is to get in touch with the people over a call to solve the quick issue as quickly as possible. Else, set up a Zoom call or Teams meet so that it can be rectified. Else, if it is not solvable at our end, then we will escalate the same to our US team, so who are on opposite time zone of ours. So in that case, it might take a little longer time. So this is the process what we follow here, and this is something about our company. And here are our partners, to name a few. The list: Chemaxon, everyone knows, which is a global leader in cheminformatics, where you can do, it's a kind like Chemdra, where you can do structure sketching, editing, various property calculations. And with regards to property calculation, I believe this is one of the best tools which you can try out. And we also have ChemPass, which is an AI-assisted forward reaction, which is based on a rule-based AI. And we also have a key module, something where if you have lots of data in documents, where you can convert those uneditable structures to an editable format. We also have something called Setara, where you can do lots of, it's a useful in the data visualization and all that. And Transgene, which is the only Indian distributor company with which we are collaborated, like, which provides electronic lab notebooks, chemical inventory, LIMS, and other compound data management systems. And coming to SCM, which is again Amsterdam Molecular Suite, AMS. Again, if you wanted to do high-end quantum calculations, DFT, HOMO, LUMO, and high-end MD, you can try using this. So, and we also distribute Molsoft. And today we'll be discussing about the PROTAC modeling, one of the tool from Molsoft. So this is again, to many of you might not have heard about Molsoft. which is again not a new company. It's a very old company which was founded in the year 1994 by Dr. Ruben Abagyan, who is still a practicing professor till today. So at that time, he developed two methodologies called internal coordinate mechanics and biased probability Monte Carlo methods. So these two technologies are the backbone and the base of this software. And you can see again the earlier adopters of this technology. BMS as well as Novartis were the earlier adopters of this particular technology. And the next decade, a number of other tools were also launched, like Scarab and ActiveICM. Again, ActiveICM is free plugin, Java plugin, where you can download and do all kinds of visualization using ActiveICM. Again, we also developed something called Novartis, 3D ligand editor in collaboration with Novartis, where this was developed with the request from medicinal chemists. Like usually what we do, computational chemists, we take out the ligand from the pocket and design new molecules and then dock them. But here you can put the docked compound or the co-crystal molecule. All the editing can be done in the ICM, in the active site itself. And this is one great advantage of this 3D ICM 3D ligand editor. And these are things like GPCR challenge. We also took part in this challenge and we were ranked first top ranked commercial tool, and we also took part. These are the few success stories. Again, we also have something called APF, atomic property fields, which was developed by Dr. Maxim Tortov, who is the CTO of Molsoft. And in this next decade, 2010 to 19, we also launched a number of other products like Molescreen, which has around 2,500 high and 2D fingerprint and 3D pharmacophoric models for drug repurposing, ADMET, and other things. We also launched Giga Search, and the other things are the success stories of ours, where you can see a company called Rampex launched a drug name called Vibomir, which is a beta-lactamase inhibitor. And they claim that this particular molecule was developed using ICM covalent docking tool. And other challenges we also took part, and other success stories are there. And in 2020, again, this particular decade, we also launched something called RIDE, which is like rapid associative discovery engine, which can screen up to 1.5 million conformers per second per GPU. Again, this is on GPU. Even on a CPU, it is not slower. It can screen up to 6,000 conformers per second on a i7 system. So we also integrated AI. We also have something called AI-based scoring function called RTCN and docking score, radial topological convolutional neural network score. And we also have certain other AI-based tools like Ridge, which is again GPU-enabled docking, which can screen up to 100 molecules per second per GPU, and GigaScreen, which is a combination of Ridge and again deep learning models, which is again GPU-enabled. And we also, ACM also been integrated with multiple other companies like Arrakis Therapeutics, Alien Therapeutics, and many more. So this is the complete landscape of ours. So Molsoft is not a new company. It's a 31-year-old company overall. So again, this is our global subset users list, where you can see Pfizer, Novartis, all big pharma companies are using Molsoft for their molecular modeling research. And coming to academics, you can see New York University, Duke, NCI, and University of Toronto. These are also the institutes which are using ICM Molsoft. And coming to Aravinas, which is, as today's topic is on Molsoft, Aravinas is one of the leading organization in developing PROTACs. Even they also are using Molsoft for their research, for their molecular modeling simulations. Coming to the Molsoft users in India, the biggest CRO in India, Syngene, also uses ICM Molsoft for their research. And we have other very good companies like PI Health Sciences, Zydus, Origin Oncology, Origin Pharma, and other companies. And from academic side, Naipur Mohali, Naipur Guwahati, Geetams, VIT, Vellore Institute of Technology, PSG, Central University of Kerala, NIRMA, and GSFC, and many more. So these are the Molsoft users in India. So what are all the modules and functionalities? We have only two, unlike other tools, we have only two modules. Like we have ICM Pro and plus VLS. So this ICM Pro, which is the base flagship model of ours, on top of that, if you have VLS, you can utilize, I mean, you can use the other tools like Chemist and Pro. Chemist Pro comes for free, but you can utilize all the functions if you have ICM Pro and VLS module. So again, these are all the few steps like hit identification and hit optimization studies, lead identification and optimization studies, which we usually carry out during the research. Like these are all the few computational techniques like virtual screening, QSAR, pharmacophoric models for pharmacophore screening, flexible docking, R group exploration, 2D, 3D QSARs, and many more. So just with these two packages like ICM Pro plus VLS, you can carry out all these techniques, computational techniques for your research. And these are all the few functionalities ICM Pro. Like you can have access to ICM Pocket Finder. Again, in docking, you have three different types, multiple types like small molecule, protein, induced fit, three different types of induced fit docking, peptide docking, RNA small molecule docking, and RNA protein docking, PROTAC modeling, which is again the part of ICM Pro, the base module. You will also be able to do MD simulations and all that. So this is again, you can generate your own 2D interactions. You can do homology modeling, protein modeling. You can do predict the effect of mutation using Mercy algorithm. You can calculate the delta G of the complex. And one good part again here is it is already integrated with multiple biological and small molecule databases where you don't need to go out of the interface for searching any downloading or searching any PDBs. Again, you can also access directly from the interface like the chemical databases like PubChem and Chembell and Drug Central and COD as well. And coming to the VLS functionalities, most of the tools with regards to Pro, there is a scalability. I mean, you cannot scale up your experiments, but if you have a VLS, like you can do ligand-based APF docking, structure-based VLS, again, fragment based. You can also do, I mean, these three, four things like covalent docking is not part of ICM Pro, and RIDE, which is not part of ICM Pro, ADMET calculations, and 2D, 3D QSAR. And these are key techniques which are not part of your VLS Pro license. These come into the VLS license. Again, this is one of the success story with regards to PROTAC. This article, application of bivalent quick approach to target aerosyl DNA phosphodiesterase on TDP1. This was published very recently in the same year, 2025, in the month of February, where NIH used PROTAC modeling tool from Molsoft for designing PROTACs onto this particular target. Otherwise, on this TDP1, this is a first-in-class PROTACs, first-in-class molecules. There were no small molecules reported on TDP1. And again, on the extreme right, you can see with regards to C alpha RMST, and this is with regards to PROTAC RMST compared to all other tools. ICM is much better with regards to RMST. Yeah, let's quickly jump on to our topic of PROTACs. What is a PROTAC and what exactly is it? The PROTAC by name it says like proteolytic targeting chimeras, right? What do? They induce the degradation of the proteins. I mean, the protein of our interest or the protein which is responsible for a specific disease. We can degrade those proteins by which using these PROTAC molecules, designing the PROTAC molecules. And this particular technology was first developed in 2001 by Crew and Deshays. And what are all the different components of a PROTAC? So it has three parts. Like first, the ligand of the target. This is usually called as POI, protein of which binds on the POI, protein of interest. It can be anything, like it can be a kinase inhibitor or whatever it can be. It might be the target, I mean the molecule of your interest, small molecule, followed by the ligand on the E3 ligase on the other side. It can be VHL or it can be cerebellum like thalidomide, lenalidomide or pomalidomide. On the other side, these two bifunctional molecules connected with a linker. Maybe it can be aliphatic or it can be pegylated linkers, and multiple linkers are available on the open databases as well. So this particular PROTAC has one side, it has the molecule of our interest followed by the E3 connected with the linker. So what is the exact mechanism of action? What it does? So initially this PROTAC goes and binds to your target protein, and then the other part of your E3 ligase, it is like once this ligand is bind to your protein of interest, then the linker along with this E3 ligase ligand, it is in, I mean, it's available free in the cytoplasm where it recruits certain specific E3 ligases which are responsible for ubiquitination. This binds and then it ubiquitinates the target protein, means the protein of our interest, and then the ubiquitinated protein is ultimately sent to proteasome where the protein is degraded and reducing its overall the expression of that protein in the at a cellular level. So what are all the advantages of doing it? Degradation versus inhibition. So it completely eliminates the function of the protein, completely eliminates the function entirely. So catalytic, one PROTAC can degrade multiple proteins as well. Once the degradation is done, so the PROTAC is again free. Once the ubiquitinate protein is available, the target protein, the protein of interest, it goes to proteasome as I was explaining here, right? Again, this PROTAC is available free for, and it again recruits, it binds to the new POI, and again it recruits a new E3 ligases and the process continues. And we can also overcome the resistance of mutations. So what is the clinical pipeline of these PROTACs? So there are more than 30 PROTACs that are there in clinical trials as of now. I think beyond that. So the companies, what are all the companies that are working? Like companies like Aravinas, BMS, Bygene, Chimera, Nurix, C4 Therapeutics, and many more. So this is just a small list again. So what are all the most prominent targets into which they are working? Like this is androgen receptor, estrogen receptor, BCA, BCL again, and BRD4. So max, like you can see, most of them are the anti-cancer targets. And is it only, and what are all the different indications on which these PROTACs research is going on, like cancer, neurodegeneration, and inflammation. So here are the few examples of two molecules from Aravinas, which is ARV-110 is being used for the targeting the prostate cancer, and ARV-471 for the breast cancer. So this is something about the general intro part and the mechanism of action, advantages, and the clinical pipeline. So let's see what are all the different the clinical statuses of different PROTACs that are there. And you can see there are three molecules like ARV-471, which we have seen, and BMS, and this is Bygene molecule. So these three molecules are in the advanced, these are in the phase 3 trials for cancer. Again, this is again for cancer, right? And again, there are multiple compounds from Aravinas in phase 2, and you can see, and even BMS. So C4 therapeutics, you can see maybe 1, 2, 3, 4, 5, 6, 7, 8, maybe 10 to 12 molecules are in phase 2 and 3 molecules in phase 1, phase 3, sorry. Phase 1, this is again a small snapshot. There are many more compounds which are there in the phase 1 as well. And these are all the few molecules which were terminated in the phase 1, because maybe due to some other lack of efficacy, or maybe due to certain other contraindications as well. Again, these are the list of PROTAC targets and phase 3 clinical compounds. How do they look? So what are all the different targets we have seen there? Like this is estrogen receptor, androgen receptor, BTK. This is again a kinase. This is again a KRAS protein, which is a mutated one. BRAF protein, IRAC. So this is again, these are all the different protein targets on which there is a lot of research going on these particular targets. The same three compounds which are there in advanced trials, you can see how are their structures. And in every molecule, you can see this particular site, the one it binds to the protein of interest. In this case, this is the one. And here also, this is the one. And these, and the other side, it is a CRBN or thalidomide or linolamide or pomalidomide. So this part, and this part, they bind to your E3 ligase. And in between these two, you see the linkers. This is a very less rotatable bonds linker. And here also it is, this is the linker, what we can assume. And here, this is the linker. So these two ligands are connected with a linker, rigid linker. Again, they can be again categorized like these are the whatever we see here are almost rigid linkers and their clinical status. Yeah. So now, how do we do this PROTAC modeling, and what are all the prerequisites that are there for for doing this modeling study? So what are all the things we need? Initially, we need the binary complexes of both the proteins, the protein of our interest. Like, let's assume if you are working on a BTK, then you may have to have a docked a molecule, small molecule. Either it can be a docked compound, or if it is a crystal structure, also well and good. But it has to be docked, and the binary complex on the other side, whatever the molecule which you wanted on the other side with the E3 ligase, like you may have to either the docked complex, docked structure of CRBN or VHL or any other E3 ligase compound. So that way you need a binary complex on the other side, right? So these two are minimum, followed by your uh, the molecule of our interest. So we need to sketch. So use these binary complexes as validated templates for ternary modeling. And the introduced designed new PROTAC, which is having the warhead and the E3 ligase. So this is what is we have seen earlier, like these two, warhead and the E3 ligase molecule, or warhead, you can also call it as a POI. So these two molecules are connected by a linker, which is introduced. And what it does, it uses the binary complex and it docks first the molecule, the complete PROTAC, then onto the protein of interest. And it aligns to your ligand, which is there in your binary complex. Then similarly, it aligns the other side of your molecule onto your E3 ligase. And then it generates, the linker is optimized using Monte Carlo simulations. So this is what it does. It generates multiple conformations of that. And then it also allows the side chain flexibility during this simulation to avoid the steric clashes. And then it generates a ensemble, means a number of conformations are generated, which are of low energy ternary conformations, then which are again rescored based on certain parameters. So this is the overall methodology or the process which takes place during the docking process. And to see how does it look again in the tool. Again, this is a very straightforward approach where this is an input where you give your target object is our protein of interest, where you can see. Again, this is a ligase object, right? And this is the table of PROTACs. And you need, what you need, all you need is the 2D structure of the PROTAC which you wanted to dock onto these two. Again, your ligand, whichever you wanted to dock, or the PROTAC. So it must have uh, the bifunctional sides. So it should have the moiety of the target object, also the ligand here also. So they know with that kind of design you should have. And this is molecule index is something where if you, let's assume like in a 2D table, you have 10 or 20 PROTACs. So let's assume if you wanted to dock 10th PROTAC, then this is again the same thing, the number. This is what you can choose the 10th number 10 molecule, and you can dock that molecule. Effort, again, effort is something which tells you about how intense you wanted to run this simulation. So this is again effort of 1 on number of parallel 5 runs is good enough for this. If at all you wanted to increase the effort to 2, you can do that again, but it takes much longer time to complete this simulation. And again, overall it takes approximately 2 to 3 hours to finish the one PROTAC docking job. Again, it again depends on your uh, system features, right? And this is the input, which is very simple, like, right? And on the right side you have how the output looks. So once the job is finished, you will see a stack of PROTAC molecules all. So this will generate all the confirmations, and the confirmations are stacked, multiple confirmations into a one particular complex. We will look into this in during my after this presentation. We will look into this how to analyze them also. And these are all the different scoring parameters which it gives. So what are all those scoring functions and how do they vary? So it ranks based on the rank order. The first column, which is I, is a rank order based on the lowest energy. And this gives an energy, ranks them 1 to whatever the number it might be. That is again based on energy, the total energy of the bifunction, or the ternary complex, the dot and everything. So RMSD, if is something in this above structure, here you can see the reference structure is no reference. Like if you already have if you are aware of the wanted, like if you have a co-crystal of a PROTAC, then you wanted to see whether is it reproducing that, you can definitely try giving it in this particular fashion. Like the first would be your PDB name, and into which that PROTAC is being chain B of that particular PROTAC molecule, or chain A, whatever, where the PROTAC is binding, and then the ligand name from the PDB. So this will be filled up. So how much RMSD it is you are getting. So it will, you can, like once we get some values with that RMSD value, then you can easily justify like how well it is, how close it is with regards to your crystal confirmation. This is something comments where the user defined comments can be added. Again, EY is the ICM force field energy. This is a surface energy term. And van der Waals, these are the van der Waals interactions. Again, energy, electrostatic energy. So these are the different scoring parameters. And delta surf is something, is a solvent accessible surface area between the two proteins, and energy of the side chains as well. So these 5-6 scoring, these are the different scores what we see. So coming to the, so this was the input file, and this is the output file. How does it look? And these are all the scoring parameters. And now this is in generic. How do you analyze? So the ENER, the energy column, is the internal energy of the PROTAC conformer, which we usually consider is used for filtering out the unrealistic high energy structures. Using that, we can consider the conformations which are higher energy, we can just exclude them to some extent. But again, it doesn't guarantee that the lowest energy conformer may always look like your biological reference compound, especially more specifically with regards to PROTACs, because these are have high, I mean, very big molecules with more number of rotatable bonds. So for that reason, what are all the different other things it is suggested is to cluster and pick the uh, conformer geometries which are energetically favorable and entropically accessible. So this is, we can cluster them and take those cluster representatives. Again, the low energy conformers with dense clusters. I mean, let's assume you got 500 confirmations, from which if you are finding like maybe 50 conformers into the similar energy range. So those can be considered. That kind of clusters we can take and consider them for further analysis. Again, isolated low conformers without nearby structures may indicate a strained or rare geometries which are biologically relevant. So even though the molecules, even though the total energy might be lesser, but they may not be very much closer to your biologically relevant conformations. And again, this way, after analyzing, we can take up the 10 to 15 complexes for MD, or we can again, if let's assume like you have found like 10 different conformers with low energy, or in these particular dense clusters, we can take all those 5, 8, or 10 conformers, and you can generate the take all those 10 PROTACs as the possible biologically relevant conformations, and then we can try using all those 10 PROTAC conformations into a single grid using our 4D docking, and then you can you can dock your new design molecules around into those 10 compounds with those 10 conformations using a 4D docking, just by a single job. So this is something again 4D docking, which is a very interesting feature of Molsoft what we have. So this is something all about the presentation part. So I will quickly try to show you how to set up the job and how the result, how to analyze the results, how does it look. So now let's assume this is the 4CI1. This is the CRBN molecule of the E3 ligase. You can see here. So this is a binary again. As I told, initially we may need two chains. This is the E3 ligase on one side. Binary complex means you have a thalidomide in this particular pocket. Again, to understand, you can go to right click on that, go to display and lag and pocket interactions. This way you can analyze what are all the different hydrogen bonds this particular molecule is making with this moiety, the header. And then you can analyze the similar thing with your 3xmf, which is my BRD4 domain ligand. So this BRD4 ligand, BRD4 protein with the ligand, and 4CI1 with the ligand. These two are good enough for me to start my simulation. So these two again can be in different orientation or different plane as well, right? Once you launch your job. So let me show the setup as well. Once we have these two binary complexes in our graphical window, all you need to do is go to docking, protein docking, PROTAC modeling. So this is the target protein. Here our target protein is BRD4 domain. BRD4 protein. We will select the 3MXF and the ligase. The ligase is 4CI1 and the PROTAC. PROTAC molecule should be there in your chemical window this way. Again, here this is only one PROTAC. You can have a list of PROTACs in the same window, and this particular molecular index encodes for this number. If I have 5 PROTACs, then if I wanted to fifth PROTAC, then I can type 5, then it will take the fifth molecule and start the docking simulation. As I said, PROTAC, we will keep it by default and number of parallel runs would be 5. Even you can increase this effort to 2, but it might take a longer time and a longer simulation time. So this is just a straightforward approach as an input, as which we have seen we have seen in the presentation as well. Now, once this particular simulation is over, then how the output looks. What do we get is, this is the output what we find, right? I mean, this is the way it generates. So both the proteins into, you can see into one single this. So chain A, this is the chain B. So this is the chain B into which this particular PROTAC, what you are seeing below, is being docked. This is the PROTAC molecule, right? So as I know that this particular molecule which I docked, 6B is collected from 6BN7, where the crystal structure of 6BN7 with both BRD4 domain and the E3 ligase is available. So what I did is I tried to import this 6BN7 and I tried to align this 6BN7 with the 6BN7 with the docking PROTACS model. So now let us analyze whether is it able to predict the biological conformation. If yes, how close? Right now, let's see. I'll try to off this. And once you have this stack, so what after docking, it will generate a stack where you have multiple conformations. Like you can see when I expand this stack, this is the first conformation, which is again, this is the same table what I am saying about. Right. This is again the rank order based on energy. And as I did not give any input RMSD, so this is 0. This, if you have any comments, so this is the same. These are the same things what we discussed in the presentation. Now, based on energy, they are ranked. And now if I go to the next one here, it will load your next confirmation this way. Else, even I can click on this button for next confirmation as well. One by one, I can move and I can see. And what happened here, you can see on one side, this POI means BRD4 domain. BRD4 protein is, static. Only the E3 ligase is moving. Why? Because this particular PROTAC is generated multiple conformations, and then based on that, this particular E3 ligase is docked. So this is how it generates multiple conformations. Now, if I wanted to run the complete, I will try to run this complete simulation and let's see how it looks. I'll try to zoom out and see. I'll click on this play button. So we'll try to see all those 510 conformations. So this way, on one side, the POI, you can see the cyan color, blue color, light blue color. It is constant, and on the other side only, your E3 ligase is generating multiple conformations, and even you can see the side chains of your POI are being rotated. So these days, I mean, the chances of the side chain clashes are much lesser here. Now let's see how it looks with your crystal ligand. For that, what I did is I downloaded this 6BN7 from the PDB, and then I tried to align this with regards to the BRD4, because BRD4 is our protein of interest, right? Now let's see how does it look. So even before that, we will try to do some more analysis. We will try to, if at all I want to see what are all the hydrogen bonds this particular molecule is making, what I can do is I will give a right click, go to display and ligand pocket interactions here. So this is how this PROTAC, you can see, is making a hydrogen bond on your POI side, and on the other side also it is making hydrogen bond with this particular on this E3 ligase side, right? There is again an additional thing what you can do. You can select the PROTAC and go to tools and analyze the contact areas in between these two. One selection, right? Right. Now when I tried this, you can see it generated a table where it is making some, what is the contact, how much is my ligand in contact with the residues of this, and you can see some of those amino acids in bigger color, bigger size, the ball and stick. So these are contributing a lot of hydrophobic interactions in this with this particular PROTAC, both on the POI side as well as on the E3 ligase side. So this is how you can understand what are all the different amino acids which are contributing to the binding of this particular PROTAC. Right now, as I said, I will try to bring the 6BN7, which is a co-crystal, and then I will try to analyze like which could be the nearest conformation. So for that, what I'll do, I'll expand this conformation and I will go with the second conformation of my from the stack. Now you can see the first conformation which you can see here. So this is the original, the gray colored one is your crystal conformation. We are trying to see how are we able to generate the nearest conformation with regards to our crystal conformation. And now here the cyan color is our docked conformation. So on the right side you can see it is like pretty much not so close, but still it is pretty much very good overlay on the right side. I'll try to off our PRD4 protein here on this side. Right on this side you can see the yellow and the blue color. So this is the E3 ligase orientation. One is the one in blue color is the crystal, and the yellow one is the docked conformation. This is the second best structure. So this is how it is able to predict to some extent, though as I said it is not accurate overlay, but still to this extent it has aligned. And let's as go to other things like one by one. The third conformation, right? It is again slightly different, but still in the same area. Now, what we can also do here? So we can also calculate the RTCNN score of this RTCNN score, which is an AI-based scoring function for all these conformations, which I did. For example, now I said like we have a crystal structure. I said structure 2. is very much slightly much closer to my to the crystal conformation. So what I can do is here I will, as I said during in the presentation, that we have to change the dense clusters. So let's assume like this is the cluster which I am very much interested in. So I can select them, and now here you can see these are all the different clusters with these RTCNN scores like 2, 6, 9. Now let's see, 2 is little closer, 6 is again slightly closer, but slightly closer, and there is a huge deviation, but still sixth one, and 9 is also into, I mean, they are all in pretty much closer when it comes to PROTAC modeling. Why? Because these are all bigger complex molecules, right? Coming to 14, let's keep this molecule 14. This is very much away, but still with regards to our PROTAC molecule, it is quite closer. 15. So this way, so overall into this particular energy area, so there are multiple conformations that are where it could generate the PROTAC modeling. This way we can do this kind of analysis. Again, this is conformation 24. Now you can see many are times in the top 20, you can see many, like maybe 1, 2, 3, 4, 5, 6 conformations are there, which are pretty much closer to our crystal conformation, crystal protein. Right now, let's assume now if at all I wanted to do because every single PROTAC modeling, it takes more than 3 hours, and every single PROTAC will generate multiple conformations. So we may have to restrict that. What we can do is from this, let's assume now the way we selected, as we have a crystal here. If we don't have, then we can select the best ones from this particular energy, low energy conformations from this particular cluster, and take those 10 clusters, 10 conformations, and we can use all those 10 different conformations and dock our newly designed PROTACs into that using 4D docking to avoid time and to avoid a lot of noise in when you dock a new PROTAC. So this is how what we can do. We can even generate the 2D interaction diagrams for this molecule by selecting this PROTAC, just giving a right click, go to tools and generate 2D interaction diagram. Yes. Now this way we can understand there is a pi here, pi cation maybe, and this is the hydrogen bonds on the E3 ligase side, and there is only one particular hydrogen bond with this the POI side is this triazole ring is making. So this way we can understand about, we can generate our 2D interaction diagrams as well. And we can also, as I said, we can also calculate the RTC name score, which is the A based scoring function, which is available with us for each and every. So this scoring function has been calculated for all these 500 different conformations which it generated, right? So how do we calculate that? Just by symbol, give a right click on your stack. You can go to stack calculations. Here, even you can calculate the VLS binding score. You can calculate RTCNN. So to calculate the RTCNN score, what do we have to do is use green or orange selections for receptor and ligand. Let's assume now I wanted to calculate the RTCNN score for this after I got this output. Then what I can do, I will double click my ligand, PROTAC, and then I will convert this to an orange color. Similarly, I will select these two proteins into green, and then I will say OK. Now I have to check this per molecule. So this, when you run this, it will take some time here. Right. Then it will give you this particular table based on the RTCNN scores. Similarly, you can also calculate the free energy, delta G. For that also, similarly, you can go right click, similar to that, we give a right click, stack calculations. You can go to binding energy here. The same way, use green or orange selections for receptor and ligand, and you can click on this button. So this is the the energy delta G, which is calculated. And this is again, you can see how the energetically, which particular energetic conformation or structure which we can take with regards to the denser conformations are much better. That's the way it is understood. So this is all about the analysis part, and you can also get this contacts which we saw here. The bigger the size of the amino acid in its proximity, it is contributing a lot of hydrophobic contacts. Like this is what is all about PROTAC modeling. So with this, I would like to conclude the session.
Thank you so much, sir, for that highly valuable and detailed presentation. So now it's the time for Q&A session. And if you have any questions related to today's topic, whether about PROTAC modeling, Molsoft tools, or a simulation workflow, so please feel to type them in the chat box. To get us started, we already received a few thoughtful questions from the audience. Let's go through them one by one, sir. So, can I import externally designed molecules into Molsoft for PROTAC modeling?
Yeah, that's what. The way you do for small molecules, similarly here, what I did, I mean, I imported this uh, one PROTAC. If you have a SD file where you have multiple structures, you can just go to this file, open just like the way you do for small molecules, select the SD file, and you can just import them here as well. So yeah, it can be imported.
Okay. So the second question we received it, sir, that is, how do I visualize and analyze the interaction of PROTAC with target proteins?
We already have seen this, which I explained. I think we can just give a right click on the PROTAC and go to display pocket and say ligand pocket interactions. This will generate the, with this you can see the hydrogen bonds of the on both the sides. And even one thing I did, which I did not show, is we can also generate the receptor surface. So this way you can again see how it is, but I mean the surface of your protein as well. Yeah, this way we can generate one way, and again the other way is other things like go to tools and analyze. We also analyze the contact areas from here with the ones which are contributing a lot. Then those amino acids are shown in little bigger size of these gray colored amino acids. So this way it displays the contact amino acids.
Okay, sir. So next question is, how can Molsoft confirmation sampling tools be used to design or optimize PROTAC linker?
How can you please repeat the question?
Sure, sir. How can Molsoft confirmation sampling tools be used to design or optimize PROTAC linker?
This is actually it uses Monte Carlo simulations for confirmation generation, like the way we did here. Again, the molecule what we have chosen here is a bit little larger molecule with a big linker. If at all now I wanted to optimize this linker, where I can design, I mean decrease the size of this linker and again rerun this particular simulation, right? Then if I'm able to generate a good score and all those things, yes, that way we have to give a multiple runs for generating those linkers with I mean designing new molecules with new linkers. That way we can go forward.
Okay, sir. So next question is, sir, what are the key challenging in modeling the ternary complex and how does the software overcome?
Ternary complex is itself a very complex thing. It's a very complex mechanism. Okay, I would just like to show one small thing where I was just very recently I was trying to analyze uh, a PDB with the name, this particular PDB 6W8I, where you can see these all three different complexes, what I am showing you here, they all belong to same PDB. Let's see like C1, C2 and C3. This is the first complex, which is, you can see in this particular orientation, it is binding, and the other chain C2, you can see how different slightly on the other side. One side, it is pretty nice with your POI side. On the E3 ligase side, you can see there is a huge conformational shift in the PROTAC and your E3 conformation, E3 ligase as well. Again, similar to this, the third complex. And again, to tell you one more thing, these all three conformations which I am showing here are the crystal conformations from one PDB only. So when you have multiple conformations possible even in PDB, so definitely it is really very, very challenging, and it has to be done with very one has to be very much diligent while selecting the conformations.
Okay, sir. So
Yeah.
next question is, sir, is it possible to run multiple PROTAC in one go using Molsoft tool?
As of now, in the GUI, it is not possible. Yeah, but using terminal, you can run them. You can run it. And again, Molsoft is planning to bring this feature in the upcoming versions. Yes, Shailesh.
Okay, sir. So next question is, sir, is it possible to calculate the buried SASA?
Yes, I showed you, right? Buried SASA is one of the scoring function. Delta surf. This is a scoring function what we have. This will give you the buried solvent accessible surface area between the two protein, excluding the ligand. So this is the one of the scoring parameter what we have to calculate the buried SASA.
Okay, sir. So thank you all for the excellent questions. Your active participation always adds great value to this session. While we try to address as many queries as possible, some may remain unanswered due to time constraint. So please don't worry. Our team will review all the questions and provide detailed responses via email after the session. With that, we officially conclude today's webinar. On behalf of Advent Informatics, I extend our heartfelt gratitude to Dr. Shamshair Singh for sharing his expertise and guiding us through the innovative modeling of PROTAC using Molsoft tools. A big thank you to our wonderful audience as well for your time, curiosity, and engagement. We look we look like we look at have a we're looking forward for another webinar. And till that, have a great day ahead and goodbye.